Before we get started, I want to acknowledge that the lands within and surrounding present-day Raleigh are the traditional homelands and gathering places of many Indigenous peoples, including 8 federally and state-recognized tribes: the Kawari, the Eastern Band of Cherokee, Haliwa-Saponi, Lumbee, Meherrin, Occaneechi Band of the Saponi Nation, the Saponi, and Waccamaw-Siwan. We share an ongoing responsibility to safeguard these lands and to respect the sovereignty of the tribes and Indigenous nations residing in North Carolina. Our goals in applied ecology are to study the world around us and apply what we learn to inform and empower stewardship. But it's important to remember that long before settler scientists decided that this was important, expert knowledge and care for the world around us has been applied as an integral part of Indigenous cultures in North Carolina and around the world. Acknowledgement is a form of decolonization, dismantling existing power structures. In order to change systems of oppression, we need to first acknowledge that the system is built on oppression. North Carolina State University is a land-grant university, which means it originally received funding through the Morrill Act, federal legislation that bought or forcibly stole lands from Indigenous tribes across what is now the United States. The sale of Indigenous lands originally raised $134,920 to fund NC State. That's the equivalent of $3,810,578 today. I hope that we can continue to disrupt and dismantle colonization beyond a simple territory acknowledgement by supporting and empowering each of you as scientists.
We are celebrating 7 students' work today. We did originally have 8 listed, but Emily Babb had an offer that she couldn't refuse. She is actually spending today visiting one of the top oldest trees in existence in Eastern North Carolina. So really phenomenal and ecology related. So she gave a presentation on Monday, whichever day that was, earlier this week, to her host lab. So we are excited for Emily. We acknowledge also that Emily has also completed the research requirement. And now we get to welcome Autumn.
Hi guys, so my name's Autumn. I work with the Brain Lab over on the vet campus. And so what my project is I work with black bears here in North Carolina. And specifically what I'm doing is I am sequencing these bears' DNA to not only individualize these bears, but also validate one of my grad students' projects. But I think it's really important for us to get a little bit of background in these bears. So first, when European settlers and Native Americans came here, they sort of had a little bit of a different view than what we do currently on bears. And they use these more as a resource, which means using them for their pelt and their meat, as well as using them for common medicine. And then we started to see a shift in the late 1700s on using these bears for more of a sport. So what they end up doing was starting to use them for collecting and killing these bears, because at this point of time, black bears were pretty stable. But that jumps into the early 19th century, where we started to see mass declines of these black bear populations. And this was mainly because of 3 different factors, not only because of hunting, but also because we end up expanding agriculture by a lot here in North Carolina, which causes us to be able to invade in their lands and kill a lot of their natural resources. But also we end up doing a lot of logging, which is, of course, deforestation, killing a lot of their habitats as well, again, as a lot of their food resources. And the last big factor was because there was this thing called a chestnut blight, and this is one of their major food resources. And now they are not having this, which is causing major declines in their populations. And that sort of led into our current day management system, because these bears were dying so often to the point that we don't even have bears in the Piedmont region anymore at this point of time. And that was problematic because black bears not only helped stabilize our ecosystems here through seed dispersal and and helping population control. But they also, again, they help with population control. And this led us into saying that we need to take care of the species. And specifically, what we end up doing is labeling the species as a species of concern in the 1970s. And that led to the management unit in 1981, which determined that we do need to keep up the bears, and we made a black bear management plan. And that has helped increase our populations by a lot. But now we're running into a new problem. We are seeing a lot of human-animal interactions, and that leads us into sort of our project of how can we manage these bears. And so this led into my grad student's project. She ended up working with the NC Wildlife Resource Commission to get DNA from 2 of the 3 management units. So we do have a Piedmont unit now. So we did not get DNA from them, but we did get DNA from both the Mountain Bear Management Unit and the Coastal Bear Management Unit. And we end up getting over 400 samples of these bears and their geographic locations. And so what my grad student wanted to do was whole genome sequence 82 of these bears to be able to make population assignments. And she also wanted to see population stability and population health with these black bears. And so she ended up getting these things called SNPs. SNPs stand for single nucleotide polymorphisms, which essentially mean in a individual or a population, you can usually see an A, and that A might switch to a T or a C, again, within the individual or the populations. And so she ended up finding 877,000 different SNPs between these populations. So as you can see here, the Coastal Management Unit and the Mountain Bear Management Unit, there is a very big distinction between these 2 populations. And also within the Mountain Bear Management Unit, we also see 2 different management groups, which means that we are seeing that these bears are not interbreeding and they are pretty separate populations. And that is really interesting. But now that leads into sort of my project of I want to be able to do this without using 877,000 different SNPs to be able to tell these populations apart, because not only is that not cost efficient, but it's also not time efficient, because we don't want to have to whole genome every single bear we come across. So that led into me trying to find the 20 most informative SNPs to be able to differentiate these populations. And how I'm doing this is I am taking 20 bears, 10 from the Coastal Management Unit and 10 from the Mountain Bear Management Unit, and I am sequencing them. And yes, we do have the location of where these bears came from, which leads me into how I'm doing this. So first, I am validating my SNP. So I want to make sure that based on our whole genome sequence bears, that we do see what we're expecting with the SNPs and the primer design that I made. And how we're doing this is we're running it through PCR and then Sanger sequencing. Sanger gives us our genotype, and then we are comparing that genotype to our previous library that we had to make sure that these SNPs and those primers do work. And then what my second step is I'm running these 400 some samples and making sure that each SNP does work, and we are seeing whether these populations can be assigned to a specific geographic location, as well as individualizing these bears. And that leads into my current results. So we have sent out results for SNP 2, and out of these 22 bears, we end up seeing that 18 of them came back to us. The ones that we didn't, we would only send out the forward primers. So we're assuming the reason why the ones that didn't work was we need to send them out with the reverse primer. So we will end up sending those out after we end up confirming with the whole genome sequencing that that's what we think the problem is. And then after that, we end up getting great results. We got 10 bears from the Mountain Bear Management Unit, and within that unit, we saw 9 out of the 10 bears are getting the results we were expecting, and the only one that didn't was heterozygous, which also is not against our results because that we had a 96.7% frequency of having the expected allele that was in this SNP. for the mountain bear unit. And then for the coastal bear unit, we got back 8 different results. And within those 8 different results, 3 of them were the exact unit, the exact allele that we were expecting. And the other 5 were again heterozygous, which makes sense because it's not going into our alternate allele, which is looking great. And so that leads us into our expected results with the rest of our SNPs. So we are expecting to be able to not only individualize these bears, which could be really helpful for the management units because that can let them know, hey, what is our bear population looking like? What is, how many bears do we have in each of these units? Are these bear interbreeding? But we're also expecting them to be able to see the difference between the 2 subunits, which this is sort of showing like a PCA panel of we can separate them out. And we are hoping not only that, but we can individualize each of these bears and see each unique genotype within these bears. And that leads into sort of the future directions of this project is not only are we hoping to see if this works across state lines, because our big thing here is we have big cities in the Piedmont region, which is why we're thinking that it's separating these bear units. So could this work across state lines? Can we do this with different types of DNA? So eDNA or trace DNA, we currently aren't using, but it would be great if we could do that because that would make it a lot easier to run this panel. Can we do this with the Piedmont bears? So we are seeing a little bit more of a Piedmont bear unit, and we have only small bits of DNA from them right now. So we're hoping to get more DNA from them and see if the PCA panel runs. And if so, is this still going to be a different group than the others? And then lastly is, could we use this with different animals here in North Carolina? Because that could also help the management units. And here is my acknowledgements, and I thank you. I thank everybody that helped me through this project.
Want to open it up for questions?
Okay.
In case anybody has questions.
I maybe a basic one. How does the bear management unit, which is the term I'm not really familiar with, relate to the population there? You kind of imply that it's the same thing, but then what's the term unit management?
So the management units are the total broad. So the Natural Resource Commission are the people who are making these management units. So they subdivide them into 3 different areas. Because she made a great little graph of this. So we have 3 different units. That's just depending on which area they subdivided them out into being, which includes coastal, Piedmont, and mountain. And the reason why that is correlated to our population assignments is because that's how we were subdividing from where the bears came from. So they told us our geographic location, so whether they came from the mountain bear unit, coastal bear unit, or Piedmont unit, and then that's when we took the DNA from each of those to see the different SNPs between the populations. Yeah, so this guy will have the mountain bear unit, which are the green and yellow. So those are still part of the mountain bear unit, but we are seeing 2 subpopulations within that mountain bear unit, which is what we're hoping to be able to also differentiate. Yes.
Follow up. So is the Wildlife Resources Commission considering then splitting their existing bear management unit into 2 management units since they're genetically distinct?
I don't think so, because those bears probably would be easier, but that would be something that we would have to ask them based on our results, because right now we're just confirming those results. But my prediction is no, just because the more you subdivide, the more resources that are going to be harder to handle. But I can't speak for them as of yet, just because we are confirming this, because if SNP panel can't really work for them, I don't think it would be best to try to subdivide like that.
Yeah, a quick question. So it seems like the genetic data is working pretty well at recovering the structure, but do you work with the bear unit at all to understand, do they do some radio tracking to say, are they actually moving between these subpopulations? Are you worried that the SNP panel might miss?
So we are not radio tracking them, but also through their DNA, you can tell whether these bears are crossbreeding. So when we were initially doing the SNP panel, we did eliminate like parents and sons so that it doesn't skew a lot of our data to say, hey, this mother had this SNP and this son had this SNP. And so we were looking specifically at non-related individuals when we did the SNP panel, specifically so we could tell the difference and who's interbreeding. Yes.
Yeah, so the two mountain populations that we're identifying,
Mhm.
are they geographically distinct as well?
That would be for my grad student to answer if I'm being completely honest. I do think that there is a difference between geographic locations. A lot of it, usually, for example, in our Piedmont region, the reason why we're thinking is because big cities, which means big highways, and while male bears are usually the ones to disperse, they don't want to try to cross those major highways because that is a high risk to their population size and their breeding. So to not die, they aren't interbreeding with the different populations, and we're assuming probably the same thing is happening in the mountain bear unit.
Another question, so
Yes.
the SNPs in the original panel,
Mhm.
were they expected to be homozygous in these populations? You know, because if they're not homozygous, then how are they? How can they identify the subpopulation?
So
They are. Get heterozygous.
great question. So we were seeing that a lot of the bears were homozygous, but we were seeing that some of them were heterozygous. Like we said, we made a spreadsheet with all the different frequencies. So for example, and that's within the mountain bear unit and the coastal bear unit. So for example, in the mountain bear unit, we saw that 96.7% were homozygous for the C allele. But for ones that are heterozygous, those we were looking at whose peaks were there, as well as we do have a allele frequency, so they're more likely to express a C than a T. And we do see in that population that there is like 2%, no, 4% chance of them having that T allele. Which is why we're doing multiple SNPs, because when it gives us heterozygous results back, that does not determine specifically what unit that goes to with that one SNP, which is why we want to do 20 SNPs, because out of those 20 SNPs, that should be able to distinctly tell us which unit they're from. So yes, heterozygous might not be what we want to see back, but it should not hinder our results unless it is showing the exact opposite or alternative allele. Does that make sense?
Thank you so much.
Good morning. My name is Matthew Keough-Lopez. I did my research in the Sustainable Health Ecology Lab, and this is parasites and egg production in female snakes. So why is this important? It connects parasite-host dynamics with life history trade-offs, asking the question, if and when a host is burdened with parasites, does reproduction pay the price? It's also important that we do this research in snakes, since they are an underrepresented suborder of animals, since most of this research is usually done in either birds or mammals. For some background information on parasite-host dynamics and trade-offs, we usually see severe negative effects on parasitized hosts, usually attacking the fitness and reproduction of that animal. However, there has been research done on keelback snakes that shows that some parasite types don't really have an effect on them. So this goes into our gaps. Of course, research is very species specific. Some species will react differently to the same parasites compared to others. This also goes into different life histories. Some can be considered more fast-paced, like for the keelback, compared to other species that might be considered slower paced. Also, for looking at certain types of parasites, some of those have been missing from previous research, including pentostomes and acanthocephalans. We could also look at the difference between captive and wild research, since some previous research has been done on captive animals. However, because they have unlimited resources, those results might be different from what we would see in the wild. So to solve some of these gaps, I asked the question, is parasite presence a significant predictor of offspring after controlling for the size of the snake? With some hypothesis being that snakes infected with parasites will produce significantly fewer eggs than uninfected snakes. and that the number of eggs produced by infected snakes will differ significantly depending on what type of parasites were found inside of the snake, with a few predictions being that uninfected snakes will produce a greater number of eggs per clutch compared to infected snakes, and that the greater difference in egg production will be observed between the parasite type, causing either the most or the least harm on that snake. So for how we did this, we actually received donations from the museum and also really anyone who finds either a roadkill or dead snake. I actually brought one in a couple of weeks ago. And we keep these in a freezer in our lab. So whenever we want to collect a sample, we'll just take one out and begin our measurements. And before we even begin our dissection, we collect data on length, maturity, sex, and species. And then once we have that down, we then begin our dissection and we can look inside the snake where we'll determine if it does or doesn't have eggs, and if it does, how many it has. And then also if it does or doesn't have parasites, and then if it does, what type, how many there are, and where they're found. And then once we have all of that data collected, we can then begin our statistical analysis. But then even before that, we have to clean the raw data, which for me, it meant cutting it down to mature female snakes and then finding out how many species we had for each of those. And then also creating separate columns for each type of parasites we're looking for, creating binary code to find out whether they're present or not. And then once we have that settled, we can then begin our statistical analysis, finding p-values and creating figures. So here we have a picture of us measuring the length of a snake, and then two pictures of eggs. The top one you could see with the naked eye, and then the bottom one you would probably have to use a microscope to see. And then here is a picture of the raw data, and then a graph that I didn't end up using that just shows how many parasites we found depending on which organ they were found in. But yeah, so after we filtered everything down, we kind of just ended up using one species of snake, being the banded water snake, because that was the only one that had a sample greater than 20, with this one having 22 females. And then the types of parasites we were assessing were nematodes, trematodes, pentastomes, cestodes, and acanthocephalans. And then also for the snakes, they all had clutches with eggs between either 5 or 53 eggs. And here is a picture of a banded water snake, and then pictures of the parasites we found, which we all took in the lab. So then with all of those results, this is the figure recreated. However, since none of the p-values reached the threshold to be significant, we did not find a correlation between the two, which is still good. It just means we can check this off the list now. But yeah. So yeah, like I said, parasite burden is not associated with egg production, which is still consistent with previous research done on the keelbacks. However, we do see a trend in the cestodes, since that did have a p-value of 0.005. However, since we do only have one sample of that, it's not really enough information to make any concrete conclusions. So we would need a bit more samples for that. So for some applications, we could use this in captive breeding and wildlife management. We could also use this for disease surveillance and monitoring protocols, and also with some conservation implications. For some pitfalls and challenges, at least for me, working in our studio as a beginner was a bit complicated, since I kind of had to learn it as I was working along. Also, with our sample size, we did start off with a large sample size, but as we were filtering it down for what we were looking for specifically, it did shrink dramatically. And it's also a little difficult to look at the overall picture because of how complex it is and how many things we have to look at, which kind of goes into future research. We could start looking at parasite density and load and how many we find and how that may affect the snake. We could also look at where the parasite is found, specifically what organ it'll be found in, depending maybe the lungs or the gut. We could also expand this research into different species, since again, they might react differently to the same type of parasite. We could also move away from female snakes and into male snakes, since some parasites have been shown to attack the tissues connecting the tail to the rest of the body, which in some cases, the tail will fall off, reducing their ability to breed with other snakes. And we already have this data since we do measure the tail length of some snakes. But yeah, so for some acknowledgements, I'd like to thank Dr. Skylar Hopkins and Alex Nelson for helping me in the lab and also with a bunch of my code. And yeah, and also anyone else in the lab who helped me. But yeah, thank you. Any questions? Yeah.
So for females, it makes sense, like you have the number of eggs per clutch. Do you have an idea of like what fitness metric you would use for males?
I'm not sure since they come to us like already dead and like in different conditions. I'm not really sure how we might measure fitness, maybe just by the length of their tail. If it's shorter, maybe less fit, and then if it's longer, something like that. Yeah.
Yeah, so, you have 22 snakes in that sample.
Mhm.
Looked like some, maybe a trend that some had decreasing, I don't know.
Yeah.
Some that might be increasing, but p-values are not low enough.
Yeah.
You think that that could be an issue as to how many snakes you have?
Yeah, I'm assuming that the more samples we get, we would get a more accurate like findings, but I guess we wouldn't really know until that happens.
Yeah, how many snakes you might have.
As many as we can get.
I have another question.
Okay.
Does one snake tend to host multiple types of parasites? Or is it more common to have like only one or two?
I would have to double check that, but that is a great question. Yeah, I would have to double check that, but I can get back to you later.
Are there more than one row per snake in your data set?
I mean,
One row.
there's usually more than one room, so yeah. Yeah, okay, yeah. So then, yeah, they usually host more than one parasite. Yeah. Okay.
Why did you choose one of your possible?
That was just the one we had the most samples for. Yeah.
You have an idea about like when the snakes are getting infected with the parasites during their life history, like whether it's in their youth or in their adulthood? Could that possibly?
Yeah, looking at the length, since we did also use that as a control, we can kind of see that for the absence, most of those are shorter, so that could mean that they're younger. So they're not really getting as many parasites during that period of their life. And then as they get longer, and I'm guessing older, they do start to get parasites. So that might be good.
All right, I got one.
Okay.
Sorry, I just thought of this. Is there any reason to suspect that like a wealth of these parasites jacks with their behavior such that when you're picking roadkill snakes, you're biasing your sample towards like more?
That could be a good question too.
Parasites or higher parasite loads?
I'm not really sure. I'd have to look at the specific parasite types and how they would affect their behavior, but that's a good question, yeah, to look into for future research. Yeah.
It'd be cool. I guess most of those are cycling parasites.
Yeah.
You can't find them without smushing the snake tissues, right? So it's not something you could pick up when you're going through people's urine.
See, Alex, it's the beds you should have. Going to fall to my work and working on it. I don't know yet though, that it's coming.
And I guess finally, did you ever just sum the number of positives and maybe this snake has 5 parasites, this snake has 1, and see if there's any clutch size differences there rather than just presence absence of each individual?
Yeah, I think we did that for species, and I made a graph for that somewhere, but it's somewhere
Lost
by
the
level.
data mining
Yeah,
process.
yeah.
All right.
All right, thank you.
Thank you, Matt.
Okay. Wait a second. Okay. Hello, I'm here to talk about kind of like the taxonomy and distribution of wiregrass known as Aristida stricta. So Aristida stricta is a wiregrass found in the, I'd say, fan favorite sandhill pines with the, dominated by Pinus palustris, nice open canopy, lots of diversity. And so why am I kind of looking at this grass? Well, there's this kind of like interesting distributional pattern you can see here between Aristida stricta and Aristida bayrichiana, both of which are in this like southeastern North America clade that hasn't really been like looked at, which is interesting considering it is a very important grass. It is kind of like the engineer of the ecosystem because it catches fire and spreads that fire really well in the pine savannas, thus keeping the, deciduous trees in fire traps, allowing the open canopy to persist. So there's been kind of, this weird thing between this distribution where it seems like there are these two species, but they've been kind of on and off, like if they are actually distinct or not. But this kind of phytogeographic pattern that you can see where there's like kind of this southern versus northern population has also been kind of documented with many coastal plain endemics by Sori Weekly. And so like morphologically, these two grasses are pretty similar, but their main distinction is the pubescence. So like Aristida bayrichiana has like this dense like bearding along the collar, whereas Aristida stricta has like these finer hairs that go up the back of the leaf sheath itself. And so kind of wanted to determine, or at least start to determine, if Aristida stricta and Aristida bayrichiana are actually two different species with likely the same niche, or if they are the same species but with that odd like disjunct of distribution. And so to look at that, I'll be trying to find if there are significant differences between their environmental niches or if there are possible limitations to dispersal. So to do that, I learned how to use R this semester, and I can corroborate it's kind of difficult when you don't know what's happening at first. So that's kind of mainly what I'm going to be focusing on here with my methods, is everything that went into, actually learning R and creating a species distribution model for Aristida stricta. Technically, I did this for the entire clade, but the focus was currently on Aristida stricta versus bayrichiana. So as made a cute little thing, since, wasn't in the field, this is my picture of me in the field, essentially, just kind of going over a few of the evaluation points. I'll go over that later, though. So first, I had to get the data. But to do this, I downloaded species data from GBIF, which is, I don't know how many people have used GBIF before, but it's an open access database with species occurrence data from places like museums and herbaria and even iNaturalist. And then absence data, you also need, people don't really record absence data though, so I had to generate my own absence data using a method of proportional stratification, meaning that I ended up, after filtering, with like about 188 presence points, occurrence points, so I had to make 188 absence points. And then they kind of fall like equally among these folds, which I'll kind of explain more later, but basically each of these colors, like all the pinks, all the reds, all the yellows, each region has like a similar set of environmental variables. And those environmental variables I also downloaded from various places like WorldClim to get precipitation and climate. I looked at fire occurrence data because Aristida stricta is also a pyrogenic flowering species. It needs the fire to produce their like viable seeds. Then I also looked at soil layers because I just thought it would be interesting initially at first, but it actually turns out is a little more important than we initially thought. So to construct this, I did what was called a random forest, in which basically I gave it a subset of my data, and it basically created many like sub-decision trees, decision trees, to kind of come to their own conclusion. And then I guess my answer was the majority rules. And I also used this to pick like the 8 most important variables, which you can see here, in which I tried to, get some from like all 3 of those types of environmental data as I downloaded. So fire, soil, and climate precipitation. And then to evaluate it, I needed to know if my model actually worked, first of all. So I did what is known as a like receiver operator curve, in which that includes the area under the curve and the true scale statistic. So basically, I only took things that were an area under the curve that was greater than 70%, just to, make sure it actually worked. So on here, you can see like the dotted line, the 1-to-1 line. Basically, what I have here is good because positive, it is above the line, which means it is predicting, like my model is accurately predicting the presence and absence data. Basically just accurately, it's actually able to predict it based on all the environmental data that I had given it. And so, yeah, over there I just have, otherwise, like Arista districta, I had 0.86. This is this graph. And then Bayrichiana, I had 0.88. So we're in good shape that my model actually knows what it's trying to make, knows what it's looking at. And so from all of this, I got my little cute little pictures. So these are the species distribution maps that I ended up creating, again, for Arista districta and Arista bayrichiana, showing their habitat suitability, but just to make it kind of a little easier to actually look at what's going on, I just made it into 3 colors based on like a 50% threshold. So 50% suitability threshold. So basically what we can see here is that there does seem to be an actual like distinction between their habitat suitability, which would imply that there is some difference like between these 2 species that would actually make them 2 species. Because we can see that Arista districta is a lot more north compared to Arista bayrichiana. I mean, they both can go south, but what this is kind of showing is there's something happening with bayrichiana that is preventing it from moving up into Arista districta's area. And so another quick aside, want to also introduce Arista purpurescens, another of the clade. This is just kind of this is more of a generalist species just to kind of show, I mean, you can see it's significantly more distributed compared to the other 2, but this is just kind of to help you guys look at these next couple overlap density charts I made. So what these are, again, with each of my variables, I just kind of wanted to see, hey, is there like any specific, like one variable that is possibly like contributing to this like unusual distribution? So like to kind of look at these, I'm going to use fire intensity as an example. I want to talk more about the fire later, but to read this, you can see that, so the x-axis is the value of the variable versus the y, which is frequency of presence. So for fire intensity, all 3 of the species, their frequency of presence was where there was basically no fire, which was interesting. But and you can see that there are a couple random variables, like for example, the nitrogen content in the middle. It shows that like bayrichiana possibly in like 0 to 5 centimeters deep, the more nitrogen content there, and like shows that bayrichiana prefers that. But overall, the kind of like big picture of this is there's not one variable that's like truly directing their like niche that's broadly shared, like even among like the 2 specialists and the generalist species. Also did niche evaluation statistics using Schoner's D, Warren's I, and Hellender distance. And kind of just to point out, the reds are Arista stricta versus bayrichiana, and the purples are Arista stricta versus purpurescens. And so these first 2, you can see that the little thing that the closer to 1, the more similar their niches are. And so we have 0.75 and 0.62. Those are both quite similar. Among the entire clade, everything is quite similar, which is kind of telling us that there is just in general high niche overlap across the clade, not even like just between Arista bayrichiana and stricta. So like there is a lot, the clade itself has like a very high, I just forgot the word. Anyways, they all have a very similar niche. And so, yeah, what does this mean? Back to the goals. Are Arista stricta and Arista bayrichiana 2 different species with the same niche, or same species but with an unusual distribution? Still don't know. Basically is the kind of short of it. So are there significant differences between the environmental niches? No. There's like a couple random like things, like it looked like Arista stricta maybe preferred a bit more clay compared to bayrichiana, but all 3 of the species that we looked at liked clay. So in general, yeah, we don't know yet. We're still kind of looking. And then limitations to dispersal. This has been something we've been trying to figure out, which is kind of initially why we started looking at the clay, or the soils itself, is like, okay, well, maybe in Georgia, we know that there's like granite outcropping. Is that like preventing it from moving up? So again, we still don't actually know what the limitation is, but if it's an actual like physical limitation or if it's possibly just like a taxonomic like limitation, basically like, if you find wiregrass above like X city or something, you just automatically are defining it as Aristida stricta versus Aristida bayrickiana. So challenges during this, again, like I said, I was looking at the majority of the clade, but quite a few of them, so like there's Aristida stricta, or sorry, Morii and gyrans, their AUCs were just like too low. I mean, you can see in this graph compared to the one earlier, it was really having troubles actually determining presence and absence data. But more importantly, though, was the fire intensity. We were really expecting that to have a large impact given the nature of the grass itself, with being pyrogenic. That's how, I mean, it can still vegetatively spread, but you still need to disperse seeds at some point, right? So we don't know yet if this was just like a limitation of the model I created, or if it was possibly the data that we downloaded from the MODIS Terra satellite. This is something we're going to look further into in the future, because we're pretty determined that it should be a bigger, like it should have more of a, like it should be much higher, but and so like applications in future, learning about reproducibility this semester, so made a GitHub account to put all my scripts into, and which I've started a little bit, but I'm going to continue looking at future climate models to see how that also would affect the two species. Again, we're tentatively just actually calling it two species, but if there is an actual difference between, like can Aristida bayrickiana move up with the climate changing, things along that line. We are still trying to look into the limiting factors. Again, this is like a common like phytogeographic pattern that has been seen, but the main thing that I think that will actually tell us if these are truly two species is with genetic analysis, which is future, like one of the future plans is to see like polyploidy, like if they are like actually two different, like if one's tetraploid and one is diploid, then obviously they're two species. They can't do anything together anymore. And common garden is also something that we're going to look into for like assessing morphology. I thank Dr. Tiley for teaching me how to use R and learning about grass. Grass is cool. And are there any questions?
Because these are both like sort of, or again, if they are two species, they would both be like pyrogenic species. And, of course, for stricta, I noticed a lot of like the sort of overlap with the range of, for example, like Croatan National Forest. I can imagine in Florida, there's also like sort of human managed areas. Is there a thought that, like, for example, changing fire regimes or possibly even like homogenization of fire regimes might be like putting pressure on these species to either like more differentiate themselves or like even possibly become more similar?
A good question.
I also have a second question, which is sort of related about, like, for example, in Florida, where we see a lot of species overlap. Are there any like dwarfs that are seen between the two species?
So there is another Aristida species, Rhizomophora, which is currently it's thought to be like a maybe like subspecies or like more closely related to stricta. But we're also kind of wondering, because I couldn't actually even make a model of that because there's so little occurrence data itself that like I just couldn't do it. So like, I mean, there could be a possibility that maybe that is some sort of, cross between bayrickiana and stricta. But again, once we actually like get the genetic analysis of this, we will truly be able to determine. But to like your first question, like, fire, I know that in general, like we're trying to actually bring a lot of fire back because, we stopped it. But so honestly, it could have possibly like started to change. I mean, obviously like these things take a long time, so I'm not sure exactly how long it would take like for this grass to adapt to being less like to like create viable seeds, but without fire. But it is a very interesting thought. Yeah.
Your grass is beautiful.
Thanks.
Who eats this grass?
eats. Yeah, nobody really. So possibly when it's young and like, more a little softer, a little more palatable, I'm sure like it will just be grazed on. But generally speaking, when you see it, it's in this like crispy brown state. It's got a lot like it's nobody wants to eat it. Like it'd just be like eating a bunch of needles, I assume. So it's really its main purpose is to be that fire spreader to again like just not completely destroy, but keep the canopy open. Yeah. Yeah.
So these models, are they, is the attempt to use these models to distinguish the species, or is it just to characterize the species?
So, and the kind of idea is to like at least long term to kind of distinguish them and see if they actually are distinguishable. These were just these models that I created were used to kind of at least start to explore like the environmental variables behind them. Sorry, yeah.
So then, do you look to see to what extent fire intensity was correlated with some of these other variables and how to eliminate correlation effect?
Yeah, so actually like in the variable importance I like I ran like a lot of different variations between them, like between different variables. And it was always so I had 3 fire variables from the MODIS satellite, and the 3 of them in general weren't very correlated, and there was like 0 correlation basically between the fire and like any of the soil or climate precipitation variables. But again, not 100% because this is from the satellites. Like have to look into like the actual tree cover to see if that is actually impacting, because these are also low-lying fires, like the entire canopy isn't going up in flames. So we might just need more like direct like actual evidence or like data from these localities to actually distinguish, because there could be actually like a higher correlation. We just actually can't see it.
You also mentioned like data limitations. How would you, maybe not you, but like how would like science as a whole like get more data? Like the data that you would need to like run more models. Like what is missing or like needed?
That is a good question. I'm honestly not 100% sure. I mean, and maybe more like I just kind of mentioned, like more actual, like specific to the area, like, very particular data gathering as opposed to just like the very broad overview, like again, satellite type data, like that's very broad. We like chose like the 30-second arc. I think that's what it's called. So like we chose like the finest variable essentially, but it still might just be too broad to kind of see these differences. So, I mean, maybe it could be something where, like we're collecting our own data over like the course of a few years to actually like get like the really nitty gritty of the variables and see if they actually have different correlations.
What about like I guess big classes themselves? Like you said, like some of them just like really rare occurrence data. Is that just because they're really rare, or that's because we're just not surveying enough?
Again, that was a good question. It's like, I mean, in general, with the occurrence data that I have, it is quite biased towards like places that people can get to, because I mean, again, a lot of the data does come from iNaturalist. People go on iNaturalist and like, oh yeah, this is here. Let me go to that exact same place too. So there might be a lot more area in which like, yeah, we aren't actually looking, like we just actually straight up don't see it because we just aren't actually going there and recording, or people that do go there and record just aren't giving it out publicly. So yeah, it would be interesting to like actually go through again more like personally and actually find these things. But like things like Rhizomophora, we aren't even really sure if that is its own species. Like Dr. Tiley kind of has a hunch that it might just be like one of the other ones, and people just, I don't know, people in Florida just want to feel special and they call it something different. But so yeah, that's and then other ones like I didn't kind of show this. That's like this little thing. So like when I was talking about the clay, these are the ones that we were focusing on. So like you can see like the Rhizomophora and stricta Again, kind of maybe if rhizoma 4 actually exists, it's more close, it seems tentatively more closely related to stricta, but in general, so like the condensata at the top there, it is listed on, was it the red list or the North? Yeah, natural heritage list. So like that one does just generally have like lower, it just isn't around as much, whereas like Morii and Gyrans possibly like they just aren't recorded, but it is more abundant. But yeah.
Thank you so
Yeah,
much.
thank you.
Hi, good morning, everyone. I'd like to present to you my research that I did with the Zhang Lab at the Center for Marine Science and Technology this spring with under her mentorship and also under the mentorship of her PhD candidate, Lindsay Curl. I was looking at snails and seagrass and seeing could epiphytal snail grazing modulate seagrass growth in these estuarine ecosystems. So just to give a little bit of background about seagrasses in North Carolina, we have 3 different species of seagrasses that grow here. And we care about these seagrasses to begin with because the meadows that they form are highly ecologically important areas, providing great habitat space for young crustacean and fish species, as well as being fabulous nutrient cyclers in these highly productive North Carolinian estuarine ecosystems. So we have the 3 species that occur here in North Carolina are Zostra marina, otherwise known as eelgrass, of which North Carolina is the southernmost boundary. We also have the Haliduli rupia complex, of which Haliduli wrightii, which is really the species that for practical reasons I ended up zeroing in on for this research. It's shoalgrass. North Carolina used to be the northernmost boundary, though it has been seen as far as the Chesapeake Bay now. And we also have Rupia maritima, which is a species that's highly difficult to tell apart from Haliduli in terms of like practical in situ distinguishing. You really need to be under a scope to see the differences, but it's going to be more of a brackish seagrass. So these, again, were the seagrasses that I was looking at, and their growth, or go into a little bit of just sort of life story on the life history on the Haliduli wrightii, which again was the species that I looked at, is very rarely seen to be sexually reproductive here in North Carolina. In fact, there was a paper published just last November about the first time sexual reproduction has been seen in this species since 1992. So of course, spread sexually reproductively here very rarely. Mostly it's going to be spread through fragmentation, but can in theory grow to these fabulous meadows, though is uncommonly seen. Kind of interested to see why it might be so uncommon, what factors might be sort of limiting its growth here. And to look at some of these factors, I also was looking at mud snails. So these mud snails, such as Tricha obsoleta and Frontis vibex, are going to be preferentially choosing to lay their eggs on this seagrass. And then after hatching and then immediately post metamorphosis, these snails are going to be grazing on epibiota, like essentially just epibiomass that's found on these seagrasses, whether it's epiphytes or whether it's algae. And then, of course, will grow up to be adults and then graze on the substrate. So like I alluded to, there's also going to be this sort of third confounding factor here of obscuring epiphytes and other epiphytal biomass on this seagrass, which has been shown in the literature to limit seagrass growth. So this is an example of epiphytes, which is just kind of any like microscopic to slightly macroscopic material that's found on seagrass blades, as well as macroalgae. And it just limits light from reaching the seagrass, thus limiting its growth. And then I also have just here another species. This is a grass cerith, otherwise known as Bitiolum varium, that grazes on all of this material throughout the course of its life. So like I said, this epibiota is really encouraged to grow by nutrient loading as well as light availability. And then it uses the seagrass as a structure and provides food for snails, but does unfortunately impact seagrass growth. So my research was really interested in putting these ecological interactions all together. Of course, when I'm talking about nutrient loading, there's a potential that this is being impacted by runoff from agriculture. There's a lot of agriculture, of course, in eastern North Carolina. There's also boat traffic, which has been shown to impact seagrass meadows and also stirs up turbidity, which might be less impactful to these epiphytal concentrations and more impactful to seagrass.
but there's a potential that snails could be able to modulate this effect by grazing on this epibiota and modulating the effects here. So I was really just interested in doing an observational study to really see what I see, which led me to the following research questions, which are sort of pairwise. I wanted to look at, does snail abundance impact seagrass outcomes? Does epiphyte and algae presence impact seagrass outcomes? And does snail abundance impact algae and epiphyte presence? So led to my development of my protocol, which I worked on with my mentors. I wanted to select 3 different sites in and around Morehead City, North Carolina. For practical reasons, that's where CMAST is. But I wanted to select 3 sites that were all anthropologically different. And I wanted to establish a 25-meter transect at each site, along which I would sample every 5 meters using just a quadrat. And within this quadrat, I would collect data on snail abundance by species, seagrass outcomes, looking at shoot count, shoot lengths, and then also eyeballing measurements of percent cover of algae and epiphytes. And I would visit each site at low tide each week and just collect water quality data as well as all the aforementioned data at each week. So for my first site, I have Gale Shore Circle, which is just a street off of Highway 24 in eastern North Carolina. It's to the east of Broad Creek, which is just a small city to the west of Morehead City. This is in Bogue Sound. It adjoins new residential development. There's a new dock here. There's high boat traffic. And the seagrass meadow here is present, but it's a very patchy meadow. So I picked this as like a very highly anthropogenically impacted site. Lots of potential for like boat traffic, lots of potential for runoff from this new residential development here. For my second site, I wanted to get one that was a little more protected. So this is Pine Knoll Shores Aquarium. This is the research area at the aquarium, also in Bogue Sound. There's very little nearby boat traffic, but it is a very shallow area, and it can be even potentially exposed entirely, which you can see here in one of my field photos, if the tide permits. But the meadow, as you can see here, is a little more connected. There's a higher degree of habitat connectivity there for that grass. And for my third site, I was looking at Harker's Island Bridge, which is at the mouth of the North River, and it adjoins this high-use boat ramp as well as a vehicle bridge. But there's also a high potential for downstream runoff here from agriculture that's upstream in the North River. But it's also a deep site, and habitat conditions here are good for a very broad seagrass meadow. So in terms of my actual outcomes that I found here, my results. So I was looking at, for seagrass outcomes, I was looking at both shoot density as well as shoot length, which are both accepted metrics within the literature of looking at seagrass outcomes. So for shoot density, I was averaging a count of Haliduli radii within my quadrats, which I then averaged across the quadrats to give, that's the site. And then of course, that was my 6-week survey. So I analyzed this data with a negative binomial general linear mixed model in order to sort of my data was a little bit over dispersed, which is why I picked the negative binomial model. And with this model, I did see that there was sort of an impact of the abundance of the Betolium varium, the B varium snail, on seagrass density. But then when I looked at seagrass length, which I determined by using an average of 5 randomly selected shoots of Haliduli radii, I did not find any significant association with snail abundance and this length data, which really just kind of showed me that if the grass is present, the length of the grass is not being modulated by these snails. But I wanted to unpack a little bit more the significant relationship that I did find. So my Harker's Island Bridge site was most likely the cause for this significance. So as you see here, so I apologize for the kind of ugly graph here, but the bars represent the abundance of the B varium snail, and the trend line here represents the sort of the seagrass density data. So there's a slight like decrease in the seagrass density as the snail increases in abundance. But to be honest, my time series is pretty small, and the trend is only slight. So even though the p-value was significant, I have my doubts on whether this was really a significant relationship. I think it possibly could have just been environmental noise. My second research question, looking at seagrass outcomes versus epiphytic biomass. And again, this is a relationship that has been shown in the literature to be a significant one, a significantly negative relationship. I was looking at seagrass percent cover, of which shoot density is a useful proxy for this, as well as shoot length. So for my percent cover, I analyzed with a zero inflated beta generalized linear mixed model because I had a lot of zero counts or just zeros in this data in general because I was looking at percent cover. I did not find in either percent cover or shoot length that that this epiphytic biomass, whether it was epiphytes themselves or macroalgae, I did not find that this presence had any kind of impact on shoot outcomes, which again was very interesting to me. I was very surprised to see that because of course this is going to be material that's obscuring light access from this grass and potentially limiting its growth. And for my third research question, I was just looking, oops, actually that's kind of what I just talked about. For my third research question, I was really just looking at epiphyte percent cover versus snail abundance. So this is just kind of unpacking the relationship, like are snails actually grazing on this stuff to the extent that it really decreases its percent cover. So I was, again, looking at this because it was percent cover data with a lot of zeros with a zero inflated beta general linear mixed model. And I found that the mud snail Frontis vivax abundance had a very close to a statistically significant relationship with epiphyte percent cover, but it was not, I mean, it didn't cross the sort of alpha equals 0.5 or 0.05 threshold, but it got very close. So I did kind of wanted to unpack it with just a little bit of a graphical analysis here. So the blue bars here are the abundance of the Frontis vivax species. And you can see kind of like a rough change in my Gale Shore Circle site as well as my Harkers Island Bridge site with changing epiphytal percent cover. But like with the seagrass density, I kind of feel like this is a relationship that can be just described as environmental noise. Which leads to my overall conclusions of this research. I found that I really sort of think that there's potential interactions between the B. varium sereth snail and shoot density, as well as potential interactions between this mud snail species P. vivax and epiphyte percent cover that might warrant further exploration or might even be potentially like explained away by further factors that I could understand with a longer time series. And one interesting thing that I was really interested in pursuing further is this lack of observation of any kind of effect between obscuring epiphytal biomass and the seagrass outcomes itself. Again, because this is the relationship that's really been seen in the literature. So I was very surprised to not see that this is a statistically significant relationship in my own research. And honestly, one thing that I'm interested in exploring there is whether it could be possible that snails are modulating enough that what we do see there, we actually, it's limited enough that we don't see it impacting the seagrass outcomes. But really, I would also have liked to have researched this during Haliduli rhytii as well as epiphytes themselves most productive season, which is going to be the summer months between May and August. So like I sort of indicated, there's some challenges with my research in terms of time series. It's the CMAS semester, so I'm just there for the kind of winter to spring, but this doesn't really overlap with productive seasons of either microepiphytes or the grasses themselves. I would love to have extended this time series over the course of the summer or even an entire year. I would love to have more intra-site replication, even like 2 transects that are 50 meters long, sampled every 5 meters, or really what I perceive would be the gold standards is being able to model these sites in GIS and then randomly select quadrats. I did take water quality data that I was hoping to be able to use to classify anthropogenic impact, but unfortunately I did not have either the time or the really like we were analyzing this data with YSI probes. I didn't have the more high-tech YSI probe that had the ability to analyze dissolved nitrogen and dissolved phosphorus, which the literature shows as impacting both grass outcomes as well as epiphyte growth. So I would have loved to have been able to use that as well as analyze turbidity data further, which again, the literature shows that the water being more turbid, less light, less epiphytes, potentially lessened grass outcomes as well. But then I just also wanted to look at more biomass and of the epiphytal biomass and just say like, let's get something better than just an eyeball measurement. Let's get some real numbers tied to this. But those are just some challenges in terms of like potential confounding metrics of my conclusions. And I'd like to also, of course, extend an acknowledgement to my mentors, Dr. Zhang and Ms. Lindsay Curl. as well as the folks that helped me out, Sarah Wall, the lab tech in the Zhang lab, as well as my fellow CMAS student. That's a collaborator in the Zhang lab, the work study student, Michaela Henninger, and just CMAS program in general, as well as Dr. McKinney for being a great applied ecology advisor. So thank you for listening. Any questions?
Do snail populations also vary seasonally?
That's a great question. I would love to pick that apart further. I do know that snail-like sort of, so I'm going to answer that question sort of by proxy. Snails lay their eggs. It's thought that it's modulated by like odors that they're picking up in the water, and snails kind of lay their eggs starting around now. I know that there were mud snails laying eggs in the area, but I didn't see any of their eggs themselves. But definitely because their populations are going to be hatching around like the sort of late spring to early summertime, I would definitely say that their abundance in these meadows is going to be highest during that sort of months of peak productivity. Another sort of like regional abundance thing that I found was really interesting is that I actually found a lot of these mud snails themselves not in my plots. They were like they were around. They just weren't in my quadrat spots. So my data is like kind of limited just by the fact that like I saw them, but they weren't in my little square, so I can't count them. But so they definitely might be limited in terms of like, I think that, so I mentioned depth in some of my slides, and I definitely think that some of these snails, once they reach adulthood, are going to be kind of like beelining it for the more shallow areas, especially the T. obsoleta species, which I actually started this research. Dr. Zhang was like, hey, I'm really interested in picking apart this relationship with specifically this species, but I didn't find a single Trisha obsoleta snail in any of my plots and any of my research. I just found them really close to the shorelines, but not in my plots themselves. So definitely like sort of regional, or even like I would even say like local regional within a single area, preferential habitat use by these snails. So.
I'm gonna like preface this with the reason I'm asking the question, but like bigger snails eat more. And we know that like lots of snails are in like the tributaries, and that makes them bigger, and then they eat more. So I was wondering, did you get a chance to like measure snail size at all, or do you know if any of your
I would have loved to have measured snail size. So one thing that I should have mentioned is that the mud snails are probably about, I would say like the mud snails can get between, I'd say like 1 to 2 centimeters long on average, but the grass cerith that I found, the Beavarium snail, is I would say maximally 4 millimeters long. So it's definitely possible that larger snails, even like I would say like probably whelks, are consuming those smaller species. There's probably not a coincidental in terms of like the mud snails, because they're like really a micro eater. They're going to be mostly filter feeding on the substrate. I don't think there's like sort of between the snail species that I observed, they're probably not consuming each other. I don't know. I would definitely be interested in seeing like how presence of like other snails, like larger snail species consuming these snails, has an overall impact on the outcomes.
So your study is observational.
Yes.
If you were to sort of use an experimental approach, what do you think you would gain?
I would definitely think I would be able to so as I mentioned, I think there's a lot of environmental noise going on in my data. I would love, one of the things I started talking with Dr. Zhang about was a potential like experimental design for this, but I think it would be very difficult. One thing that I think I would be able to like control though is I would be able to control the water quality, essentially the turbidity, all the kind of nutrient load. I would be able to add in things myself. So really, I think I would just be able to get like cleaner data, to be honest. I think I would be able to, like, by controlling the nutrients, by controlling the light levels, and also like controlling when I know the snails are going to be there, and thus when I think the snails are going to be grazing. So for example, a real limitation of my research was that I only went out once per week, which means I'm really only looking at whether the snail is there, even just coincidentally during that one time that I examined it. So by having an experimental design, I really think that I could hone in when the snail is present and thus be able to pick apart, okay, I think this snail is probably grazing here, versus like this snail just might be there and I might be seeing the snail and making an assumption about its behavior. A great question. Thank you. Thank you.
Good morning, everybody. My name is Isley Fryar, and I worked under Dr. Cotty. And my research is observing the legacies of redlining and seeing how it affects street tree habitancy and bird biodiversity in Durham, North Carolina. So this takes a bit of background. Everybody probably is familiar with redlining, the concept of it. So this was perpetuated by the HOLC, the Homeowners Loan Associate, or the Homeowners Loan Corporation. This was created as a result of the Great Depression under FDR's New Deal. He developed this. It was intended to refinance existing loans for homes in default and also expand home buying opportunities for homeowners or people who are prospective homeowners. They designed the color-coded maps, as you see here. This is downtown Durham. And it was categorized by their riskiness to credit lenders. So red areas were no-go, of course. Blue and green were better, of course. They were meant to be based on housing and economic measures, but this was a thinly veiled way for the HOLC to discriminate against those of color. Redlined areas were often majority Black and Hispanic demographic, so they were very obviously discriminated against. And this impact of redlining has followed us well into the 21st century, as people of color are much less likely to own their homes, and it prevents them from accumulating generational wealth. So this in turn creates a positive feedback loop, preventing them from ever being able to own their own homes, and it is extremely hard to break even when these restrictions have been removed. So I wanted to see how this would affect bird biodiversity, because redlined areas experience many different things, such as they experience lower tree cover or lower tree coverage. So they have less planted trees, leading to higher average temperatures. This creates urban heat island effects. Sometimes there is a difference of over 5 degrees Fahrenheit in these areas, which is a lot. So they also have less access to green space. These parks are predominantly found in the A and B grades I found in Durham. So it's a lot harder for people to get access to these areas. And these areas also face higher environmental burdens. So they have unequal resource distribution. There is higher than average air pollution. They face waste accumulation a lot more than nicer areas. And according to researcher Ellis Soto et al., they found that historically redlined neighborhoods remain the most undersampled or urban areas for bird biodiversity, with the sampling disparity growing at about 35% to 40% over the past 20 years. So my hypothesis stated higher graded areas will have more biodiversity than redlined areas, and I predicted that grades A and B will have higher bird and tree diversity. This is just kind of like nice little span of backyard birds. So the data I used was from Triangle Bird Counts data from 2019, 2021, and 2022. Triangle Bird Count is a citizen science survey project aimed at observing songbird abundance and diversity. It was founded by Dr. Cotty at the Lab of Reconciliation Ecology at NCSU, along with his PhD student, Dr. Jin Bai now. The surveyors will choose their points within the triangle, survey them. They report on bird abundance, the species, distance from the bird, and other environmental factors such as wind and temperature. I also used Durham's tree inventory data. They have a very robust data set of all the street trees that they have planted within the past 10 years about. So it lists the date of the location, the date of planting, and the species of the tree present. And I also just used our national NDVI maps. So NDVI stands for Naturalized Difference Vegetation Index. It analyzes the so-called greenness of an area. And yeah. So this is the map. Some of the points are overlaid, like some of these have like different years on them. So they'll have like, they're kind of under them, but this is data specifically in downtown Durham. I took the entire data set and from the triangle I excluded points not within Durham, and then I combined these points with the HLOC grades to be able to analyze it better. And from this data, I was able to produce this nice graph. So as you can see, this on the y-axis, it is richness density. So the species divided by the size of each of the grades in kilometers squared. And on the x-axis, I have the HOLC grade. So as you can see, A grade is obviously a lot higher in the richness density. There was a somewhat lack of data in the 2019 year. I only had about 7 or 8 data points to go off of, but in the 2021 year and 2022 year, I was able to get a lot more points for that one. So it's definitely a stark difference between the A and B, or A, B, and D versus the C and D grade. So that was something that was really astounding to me. And furthermore, I wanted to look at tree composition. So this is the Durham Street Tree Index, or the inventory. I took all of that and I excluded all the points not within the HLC grades. And then furthermore, I built 250-meter buffers around each of the survey points just to only include the trees that were planted within that buffer. And I just wanted to see like a raw count of just like how many trees would be around these survey points in these areas. And from that, I produced this graph. So following similarly to the other one, it is very obvious that grades A, well, yeah, B not so much, but grades A were definitely a lot higher just within like raw tree count. Grade C, this was not normalized for area, so this was just like count. Grade C is the highest grade. It is 9.93 kilometers squared. Grade A is only 1.27 kilometers squared. But the fact that there is still a way higher average of trees within these areas in the A grades, when it's like, it's less, it's probably, it's still a fraction of the size of the C grade, and it still contains that much. And then D, sadly, I was not able to get as many points within D itself. They were more adjacent to D, but the fact that they still lacked that much was very astounding to me. Oh, I also did just a little, this was just like a top 5 of the most commonly planted trees. There was an obvious preference for non-native trees, of course. Crepe myrtles, trident maple, and Chinese fringe tree. Very popular trees, prized for their showiness. But they still do provide very necessary services, like common crepe myrtles. Birds will still eat the seeds. They will still nest in them. Fringe trees provide droops for birds. So they still readily use these as resources. And this is actually just a NDVI map of Durham County as a whole. I didn't overlay the HLOC graph, but you can actually just see the area where it is just by that yellow patch. However, you can see that it is still very, like, these deep greens represent like heavy vegetation. And Durham County itself is actually very, very forested, especially in the rural areas up north. But, oh yeah, and NC itself is over 60% forested, so we still have quite a bit of vegetation throughout the state. So a few of the things that I ran into challenges with the data in terms of, well, yeah, bias. There was a lot of bias in the people surveying the points. So they are randomly generated within a grid. So some points may not even be accessible. So they might be on private property. They might not have any access points for people to get into them. And I also noticed a lot of the surveyed points were generally within the A grades. So there may be a slight preference in people wanting to go to the nicer areas to sample. Invasive species flocking was another big issue. There are so many European starlings. That was just like a majority of the abundance. It was just so many of those. So they're flocking species, so they tend to aggregate in an area together. So that might skew the data a bit in terms of abundance. And a possible preference in tree planting as well. A large majority of the planted trees are non-native, so they're prized for their showy flowers and foliage. But of course, they still provide those necessary things for birds. But not native trees are preferable for our native species. And also more mature trees were planted in A and B grades. So for the planting data, it showed that trees within the A and B grades typically were well above 10 feet. Whereas in the C and D grades, you would see trees that are planted there, saplings, less than 5 feet. And a big factor they also put in there was the soil quality. It was often fair or poor. So that's any indication. It's definitely a resource disparity. So the main thing I really wanted to see was the songbird composition, and generalist species were the main thing that I saw in this. So your average cardinal, like wrens, house finches, they were among the highest seen birds. So that is, of course, because they are generalists, so they do not have as many specific habitat conditions that they need to survive. They can live in very degraded environments, and they are ubiquitous in all grades. However, they were reduced as you go down in the grade pattern. However, this does not mean that rare birds are unseen in the triangle. Just this past week, me and my friends were actually running around trying to find that dang painted bunting in Dix Park. Unsuccessful in that, but I know it's there. It was there. And we have had quite a few rare species actually pass through the triangle. We have had yellow-billed cuckoos, American redstarts, and magnolia warblers. And these birds are quite beautiful, and the fact that they still chose to come here, I appreciate them for that. Another thing that we, of course, see in urban areas is the continuous urbanization and gentrification of areas. So some of these areas that were historically included within the C and D grades have actually changed in their land, or have actually changed in the value a lot. So this is typically due to gentrification, as people of color are being pushed out and gentrified by people coming in and buying up properties, driving up prices, and forcing natives out. You can draw parallels between that and invasive species, kind of. And then, of course, the importance of urban green space. This one is a big one. So the green space in urban areas is a critical physical and mental outlet for people living in densely populated areas. A study by Wood et al. In Bradford, UK, with 12 parks, found a positive association between the biodiversity and psychological restoration of people among park users, suggesting ecological quality, not just quantity of green space, matters for mental health. So while green space is important, it is also a mix of the size and quality of it that can really benefit both humans and wildlife. So the quality directly translates into benefits for human health and well-being. So, yeah. And I want to give a big shout out to Dr. Kadi, Dr. Jinbai, who is his research student, and of course, Dr. Erin McKinney for helping me out. Thank you.
Thank you, Isley. Well, we have time for one question, and we're going to transition to the next student.
Thank you. I was wondering, is that like bird feeders? Do you have any idea of like how that like correlates with sleep number, like any of the other variables like food conditioning for birds?
Yeah, for birds, it is really hard to just find variables that really affect them just because they're so mobile. Like they can just go anywhere they want. That would be a variable that I would like to look at, seeing if bird feeders would affect their abundance and species. That is really something I would like to look at though.
Cool.
Thanks.
Hi, everyone. Thank you so much for coming out and listening to all of our talks. My research looks at how initial camera trap deployment affects detection probability and how that relates to the assumptions of constant detection probability. So first off, camera traps are a really useful tool for wildlife management and research. They give us a lot of insight into density, abundance, all these sorts of things. And it's generally kind of assumed that animals aren't really noticing the cameras. They're kind of a way for us to monitor without animals kind of noticing or reacting to them. But do they actually notice the camera traps? It kind of seems like they do. So this is what we would call attraction behavior. This is a coyote coming up to the camera, messing with it, chewing on it. And this is a type of behavior that violates that assumption of constant detection probability. Conversely, this is a coyote showing high avoidance behavior. It sees that camera and absolutely books it. It does not want to be anywhere near that camera. So this is, again, another way that constant detection probability is violated. So once again, density and abundance estimates rely on this assumption of constant detection probability, specifically relative abundance or detection rate. However, when we have attraction behavior or avoidance behavior, that violates that assumption, and it can cause artificial inflation or suppression of relative abundance and density estimates. So attraction behavior inflates those estimates. And this is a graph showing that with attraction behavior, that constant detection assumption is violated. Conversely, we have avoidance behaviors, which artificially suppress relative abundance and also violates that assumption. So my research question was, how does initial camera trap deployment actually affect detection probability, and is there enough of an effect that it violates this assumption of constant detection probability? So first off the data, I used Snapshot USA data. Snapshot is a collaborative effort with researchers all over the country. It was established by one of my PIs, Dr. Kays, and a group of other professors, and it's run through the Smithsonian. This camera trap data set was absolutely massive. It is over 5 years of camera trapping all across the country. This is our study map. You can see we have locations in almost all 50 states. Or I think actually we do have locations in all 50 states now, so which is really exciting, and even Alaska and Canada. And for this project, I actually created my own array at Hanging Rock State Park with our wildlife club, and we will be going back each year as now a wildlife club activity with setting cameras at Hanging Rock State Park. Again, this data set is absolutely massive. We had over 9,000 cameras and over 900,000 sequences. And a sequence can be thousands of images. So this is over 1 million images easily. So for this, I used a pooled generalized linear mixed model. This is the scary equation. But it breaks down into a couple simple components. This is our response variable, or our response. It is a binary detection probability per camera day. This is our key predictor variable. It is early period versus late period, and it's kind of keying into looking at those first couple days. Is detection rate changing within those first couple days of deployment? I also have 2 array level covariates in forest cover and in log mean human population, as trying to get human presence as one of our covariates. And then our deployment level covariate was trails. A lot of wildlife species are trail runners, and so we wanted to control for this as well. So the results of our model, we had 3 avoider species. This was eastern gray squirrel, eastern fox squirrel, and eastern cottontail rabbit. But the majority of species had no effect, which is really encouraging. And we trust that since we have such a massive data set, that we have enough statistical power to confidently say that there was no effect. And then we had 4 attractor species. It was striped skunk, gray fox, red fox, and white-tailed deer. So again, most species had no effect, which is really good news for us. But some species did have an effect. So now we have reached the part, okay, we know there's an effect. So what? Does it actually matter? Does it change those relative abundance indices? So we made a worst case scenario analysis. And we found that generally there was no actual realized effect on detection rate specifically. So we decided to narrow it down to looking at detection rate, which is the number of detections divided by the number of trap days. Basically, we did a worst case scenario analysis. We took our strongest avoider and our strongest attractor species, which was gray squirrel and striped skunk respectively. And we calculated the change in detection rate by having the full data set versus excluding those first 3 days. And we calculated the difference and then picked the array with the biggest difference. So our worst array with our most avoided and our most attractive species, this was the biggest change in detection rate. That is 0.0048. It is just tiny. It is not enough to actually have any impacts or mean anything basically for wildlife management and research. And for eastern gray squirrel, it was 0.0314. So again, worst case scenario, basically no change. But we wanted to look at, does the deployment length matter with this? You know, if you're only having a 2-week-long deployment, excluding those first 3 days is a much bigger deal than if you have a 2-month-long deployment. So I ran simulations, truncating those deployments, and it also had basically a non-significant difference. So this was for eastern gray squirrel, our top avoider species. With a 1-week-long deployment, we had a change of 0.102. And so this is again including versus the full data set versus excluding those first 3 days. And this is a truncated deployment of 1 week. So again, a difference of 0.1. So it's not a huge difference. And then there's a notification later. Okay, sorry about that. So then with 8 weeks long, we had a 0.031, so basically once you get to that full 2-month-long deployment, which is a typical length for a camera trap study, the differences basically were negligible. And so again, this was for our top avoider species. Our top attractor species was striped skunk. And so we would assume that with the attractor species, the excluded data set would have a lower detection rate versus the full data set, because when we exclude those first few days, if they're going to the cameras more in those first few days, that's going to artificially inflate detection rate. However, when we looked at it for a 1-week-long deployment where you're excluding those first 3 days, the difference is only 0.007. And for an 8-week-long deployment, it's 0.005. So again, basically no practical effect. Our interaction effects were really interesting as well. Our most interesting interaction effect was trails. We looked at, so a lot of wildlife species are trail runners, and typically they tended to avoid cameras on trails in the early period. Which is really interesting because perhaps there are these animals which are around new things. They're around humans, they're around human activity. They see a new object and they're going to avoid it, even more than an animal in the forest, which might feel a little more secure in their surroundings. And the 3 species that had this interaction effect were gray fox, white-tailed deer, and raccoon. So again, this is really, basically this study means really good news for wildlife researchers and managers. There's no real, our GLMM was able to pick up a change in detection probability, but there was no realized effect down the line, which is, again, really great news for wildlife managers and researchers. We don't have to change camera trap study design. We don't have to exclude those first 3 days of data to create that adjustment period. And it also provides a lot of really interesting insight into how animals perceive novel objects in their environment. I would like to thank all of my PI, Dr. Roland Kays, and the Biodiversity Lab at the museum. I have been working there for the last almost 2 years now, so I've had a lot of fun with those people there, getting to know all of them. Shout out to the College of Natural Resources. I'm an FWCB major. But yeah, thank you so much.
do you think is driving that avoidant and attractant behavior? Do you think they're seeing it? Do you think they're smelling it?
I think it's a combination of both. I think they can probably smell the human, like humans coming and setting up. You know, we're often knocking down vegetation in front of the cameras. That just spreads your scent around a lot. Also, I think they're seeing it and hearing it. The camera makes a slight shuddering noise whenever it takes a picture. And I think that the combination of new object, new smell, new sound is just, that's what creates that attraction or avoidance behavior.
Follow-up question for that. Are those species avoidant of other types of human disturbances?
You know, that's a really good question. I mean, I feel like from what I saw, the attractor species, those are generalist mesocarnivores. So it makes sense that they're a little more neophilic. They're a little more outgoing because they're generalists. They thrive on new environments. They thrive on making a home where anyone else, where other species can.
Is there anything that you could do, like, make the cameras look like, I don't know, when you could play the cameras, like, humor gloves to like produce smell that's like on the camera? Did people do that?
Not really. Usually camera trap deployments, I don't know, it's just kind of fast and dirty. You're just getting out there to set those cameras as quickly as you can. One thing I was really interested in is a lot of, and I didn't have the time to go into this, but a lot of camera trappers will bring their dogs with them when they go to set up cameras. And for me, I thought that was a really interesting thing that could potentially create even more attraction or avoidance behavior, because not only do you have a human smell, you now have a strange animal smell. So that was something I really wanted to look at, but I didn't quite have the time to.
We had a question online that had a little bit of overlap, so it might get our wheels turning in a different way. So Jordan P says, great presentation. And the question is, do you have any speculation on why certain species are more likely to be attracted versus avoidant? And they're thinking about roadkill and car accidents because there seems to be some overlap in those reasons.
Yeah, really great question. For our avoidant species, they tended to be really high prey species. They're the bottom of the food chain. We have 2 species of squirrels and a species of rabbit. Like everything eats those species. Also, we're putting I think for squirrels, we're putting cameras on their homes. They're gonna notice a camera on a tree they're trying to climb up. So I think that is why we had avoidance from those species. In terms of attraction, again, I think those are our attractive species are typically our generalists. They also are ones that tend to have, they kind of thrive off of like humans, and they can utilize a lot of human resources like trash and stuff like that.
If you had more time and effort, are there any other questions related to the stuff you want to explore?
Ooh, that's a good question. I definitely would love to explore the dogs. I think that would be really interesting. I also think going at this at a finer scale would be really interesting. You know, we looked at this at a national scale. We were trying to see overarching trends and overarching patterns. But I think really dialing down maybe to different ecoregions, different states, different cities, I think kind of fine-tuning and taking a zoom in would be a really interesting way to continue this research.
Thanks.
Yeah.
All right. Hi there, everyone. My name is Kara Vold, and today I'm going to be discussing my research in identifying patterns in parasite infection and immune function in wild water snakes. I conducted my research in the Sustainable Health Ecology Lab, mentored by Skylar Hopkins. And as a bit of background information, I just wanted to introduce that vertebrates are frequently co-infected by multiple pathogens. And it's important that we understand the interactions between these co-infecting pathogens because they can have substantial consequences both for the health of the host and for the health of broader ecosystems. And then parasites are a natural and often necessary part of ecosystems. However, problems do arise when these existing infections increase in incidence or severity. And then these impacts can also worsen with changing environmental conditions that allow these pathogens to have greater geographic range or environmental persistence, as well as these changing environmental conditions can stress the host species and lead to decreased immune function there. So in this image here, you can see when snake fungal disease, which I'll talk about in the next slide, was first reported in New Hampshire. It has drastically increased in geographic range. Now it's been reported across most of the United States. So for snake fungal disease, snakes in the United States and Canada are frequently infected by an emerging fungal pathogen, Phytomyces ophioticola. And as you can see here in these images, it can cause these crusts or ulcerative lesions across the skin of the snake. It can also lead to some facial swelling. And then unfortunately, in some rarer cases, it can lead to systemic fungal infection, which can be fatal for these snake species and lead to declines. And as I discussed in the previous slide, since it's been increasing in geographic range and prevalence, that's why it's considered an emerging infection. The other pathogen I look at in my research is the hepatozoon causing the disease hepatozoonosis. So snakes across the world are infected by a vector-borne protozoan blood parasite in the genus hepatozoon. And it's actually very interesting compared to a lot of other vector-borne parasites. Hepatozoans aren't transferred through to the vertebrate host through the bite of an arthropod when they take a blood meal. Rather, they're transmitted when the arthropod is consumed by the vertebrate host. So in the case of the hepatozoans that affect snakes, an infected mosquito is consumed by a first vertebrate host, usually a prey item for snakes like frogs or lizards or potentially rats and mice. And then that infected prey item is then consumed by the snake, and the snake becomes infected and starts the cycle all over again when mosquitoes feed on that infected snake. And the hepatozoans that affect the snake species also are related to hepatozoon canis and hepatozoon americanum, which cause disease in both wild and domestic canines. And it's important to consider, although in many snake species, the hepatozoon infections are well tolerated, some can be reported to have anemia, organ damage, or weakened immune function. And this is a concern because if these snakes are having weakened immune function as a result of this infection, that can open the door for other co-infections, such as snake fungal disease. And a recent study by Hazelrig et al. Had found that snakes infected with hepatozoans had more apparent signs of snake fungal disease as well. So that's a concern for this. And that is what the hepatozoan looks like. It's an intraerythrocytic blood parasite, meaning it's found in the red blood cell. And so it's just that little kind of banana-shaped guy there, and that is the blood of a water snake. So why is it important that we do research on these snakes? Other than I love snakes and I think they're very cute. Snakes and herpetofauna as a whole are widely understudied, and we need to do research to fill that gap, especially because snakes are so important in that they provide a lot of under-recognized ecological roles in ecosystem services. For example, as I'm sure most of us are aware, snakes consume a lot of rodents. Rodents are an important agricultural pest, lead to declines in yields, and also lead to an increase in use of rodenticides, and snakes can help reduce those issues with controlling these rodent populations. As well as rodents are vectors for disease that can affect a wide variety of animals, including humans. And so that is also a concern with snakes as well, with their ability to control those disease vectors. And then overall, the health of these host species is directly reflective of the health of ecosystems. And if these snakes are overburdened by parasites, then we may see a reduction in the ecosystem services they provide, and thus harms to ecosystem and human health. So in terms of the questions I was looking to answer with the research, as I mentioned with some of those pathogens, we can see a wide variety of presentations with these diseases, from some snakes being completely asymptomatic for the infections to others suffering severe or even fatal disease. So we want to understand, one, how widespread are these infections, but also how do they affect snake immune function, and how do those varied immune responses lead to different outcomes in infection. And then specifically with the snake fungal disease data that we have, wanted to look at how does the prevalence of a Phytomyces ophioticola differ among snakes captured in our 3 sampling locations? And then also, what is the relationship between the season of capture and the prevalence of OO? So in terms of how did we collect this data, we conducted sampling through field surveys of snakes in the genus Nerodia across 3 states. So Craig Lynn's lab in New Jersey, here in the Sustainable Health Ecology lab in North Carolina, and then David Rodriguez lab in Texas. And then what samples did we obtain when we tramped through the bush and captured these snakes, often by hand? So first of all, we got some physical exam data looking for crusts and lesions that are indicative of snake fungal disease on these snakes. And then we took a swab of the skin from every snake captured in order to send that out for PCR testing to identify the presence of snake fungal disease. And then we also draw blood from the tail of the snake and take that blood sample and create what is called a blood smear, which is a thin film of blood on that microscope glass that you can look at to identify white blood cells present in that blood sample. And there, from looking at the percentage of those white blood cells, you can look for patterns in the immune function of these snakes. So we're looking at these cells here, the lymphocytes, azurophils, heterophils, basophils, and monocytes, and then this is a red blood cell and a platelet there. And these patterns are especially important to understand how their immune function varies in response to pathogens. For example, previous research has found that snakes infected with snake fungal disease have azurophilia or elevated levels of azurophils, which is an important pattern to consider there. And then in terms of this immune function data, we still have a lot of samples to work through. So if you imagine, we're making 2 blood slides from pretty much every snake that's captured. So we have like over 1,000 probably blood samples to work through. So there is still a lot more that we need to do in order to have this final data set, work through a lot more of these slides. However, at this point, the ratios that we've obtained for these different cell types in the samples that we have looked at do show substantial variation among individual snakes. And this suggests that there's potential for variable immune response that may be linked to infection that we need to explore further in the final data set. So in terms of hepatozoans specifically, though again, we do need to work through all of those samples to get final data to look at prevalence and things like that, we have found hepatozone positive samples in all 3 of our sampling locations. And so these are those hepatozones in sample from a snake in North Carolina. This snake in New Jersey actually had such a high infection. There was 2 present in 1 red blood cell there, and then a snake in Texas as well. One thing that was interesting is thus far, Texas has had lower prevalence of the hepatozone parasite in the samples that we've looked at. However, that's something we'll need to explore again further in the final data set there. We do have snake fungal disease results from all of the snakes we've sampled through those field surveys. And in terms of the differences we see across states in prevalence, they're very different between North Carolina, New Jersey, and Texas. So 96% of the snakes we sampled in North Carolina were positive for snake fungal disease. And then over half of the snakes in New Jersey and almost a quarter of the snakes in Texas. So why are we seeing this significant difference in prevalence between these states? It's likely due to environmental differences that allow for different persistence of the fungus in the environment. For example, Texas is a lot drier of a climate than North Carolina, and our very moist, humid climate may allow for greater persistence of this fungus in the environment. It's also important to consider that for our sampling here in North Carolina, most of our snakes were actually captured at Yates Mill, kind of right down the road, and that location sees a very high prevalence of snake fungal disease, which may be affecting that data. And then in terms of across seasons, when comparing snakes captured during our field season in the fall compared to the field season in the spring, snakes are twice as likely to be infected with snake fungal disease in the spring, which is interesting and is likely related to some biological reasons. As during the springtime, snakes are emerging from their winter brumation, and that means that these snakes often have lower metabolic rates and thus decreased immune function, so they're less likely to be able to fight off this pathogen. And then the springtime also has that warm, moist climate that allows for greater environmental persistence of the fungus. So then this is just like a summary of the results there that we have thus far, is that SFD is present in all of our 3 sampling locations with different prevalence between the 3 states. And then the prevalence is twice as high in the spring compared to the fall. And then we have those hepatozone positive samples present in all of our locations with fewer positive samples in Texas. So in terms of future directions, the first thing that we will do is to continue looking through hundreds of blood sample slides to develop that final data set that we can look at those broader patterns of immune responses there. So we want to look for distinct patterns in immune response in snake fungal disease positive snakes compared to snakes without the infection. And then also to see, do snakes co-infected with both SFD and hepatozones experience unique immune responses? And then overall, I would like to compare the prevalence across state, season, and even the host species of snake for the hepatozones. Lastly, I would just like to acknowledge some members of the Shell Lab who have helped me throughout this project, Dr. Skylar Hopkins for mentoring me and also making some of the cool graphics you've seen throughout these slides. Dr. Kenzie Pereira, who is a postdoc in the lab and helped me with developing my initial methods for this project and a lot of the literature review, as well as some field work. And then PhD candidate Alex Nelson, who helped a lot teaching me about snake dissection, parasite identification, and some field work as well. And then Corey McKinstry, who's a PhD student in Shell, who was previously with the collaborator lab in Texas. So he's collected hundreds of blood samples for snakes that were useful throughout this project. So thank you, and I'd just like to open up to any questions. Yes.
Have you considered running like a CBC or other type of blood test to see how like hepatozoans could affect like organ function?
Yeah, so with the white blood cell differentials that we're performing manually, that's our like version of a CBC. As most of those machines to automatically complete CBC tests are meant for cells or blood samples that don't have nucleated red blood cells. So if you saw in those red blood cells of the snake, they have nuclei versus for like humans, dogs, cats, etc. We do not have nuclei in our red blood cells. So the machine would see something very different looking at snake blood, being that it was calibrated for mammal blood. So that's why we're doing those manual white blood cell differentials to get similar results. And we'd like to see, do snakes have issues such as like anemia or different white blood cell counts in response to having those parasitic infections. Yes.
Because snake fungal disease has such a high prevalence in the state of North Carolina, do you notice any kind of, or is this even known that there's like sort of an adaptive response of snakes in North Carolina? Have they started to like be able to adapt to like, I guess like, you know, not necessarily resist the fungus, of course, because there's a high incidence of infection, but like, for example, the worst outcomes in snakes do you see this in North Carolina, or do you see this in other states, I guess?
Yeah, that's a great question. And that's like what we're looking at, hopefully with the like immune response data as we further explore that, is what is the difference in these snakes who are experiencing this more like overt signs of snake fungal disease versus those that may test positive but have minimal symptoms. We do see snakes that do develop this more extreme form of snake fungal disease where it kind of it infects them from the outside in and like permeates to infect like their respiratory tract and things like that. So I've seen snakes treated at the veterinary school where they have to be nebulized to inhale a antifungal drug in order to hopefully treat this disease, which unfortunately there's no well-defined treatment procedure or completely effective procedure there. But there are snakes that are found throughout the state that do have this more severe form of SFD. But then we find that at Yates Mill, for example, where there's very high prevalence, most of the snakes seem to be doing pretty okay. They do, again, test positive repeatedly or have these lesions present, but they're not having the more progressive systemic fungal infection as often as we see at maybe other locations.
Great.
Thank you. Yes.
So I'm curious about the transmission cycle of that.
Yes.
Mosquitoes get it by biting the snake?
Correct. Yeah, they feed on the snake and then take it up in the blood. Yeah, the mosquitoes can actually like track under the scales of these snakes. Unfortunately, with a lot of arthropods, reptiles are not free from getting those infections. Like we see things like ticks on many reptiles, even those like bearded dragons and things that have greater like armor. And arthropods like mosquitoes as well can feed on them. Unfortunately, even though they have those scales, it's not strong enough like armor to prevent those infestations.
So they feed on the snake when it's less mobile, or?
Yeah, when these snakes are out basking, that can be a time when they're fed on. A lot of the time snakes are found out in the open, especially if they're spending time basking in the sun or out searching for prey. So there are definitely many opportunities to come in contact with mosquitoes.
Mosquitoes specialized reptile feeding?
I know there's some multiple species of like Culex mosquito that are found to feed on these snakes. And there has been some research that actually looks at how hepatozoans, specifically the snake species, may alter the feeding behavior of these mosquitoes when infecting them in order to maximize their ability to transmit it to the next prey item. So some mosquitoes that are infected with hepatozoans were found that when the parasite was developing to be able to be in that more transmissible state, it decreased the feeding behavior of those mosquitoes so that they were put at less risk of death before they were able to transmit it to the next prey item, which is pretty interesting.
Thank you so much.
Thank you.
Thank you again to our students who presented today. Your work was incredible. I hope you enjoyed answering all of the questions that you got. We certainly enjoyed asking them and learning from you. And thank you to everybody who joined us in person and online. Happy Wednesday, y'all. Happy last week of classes. Take good care.